Welcome to Mountain Voices, a monthly celebration of all things White Mountains and Lakes region. This year, Mountain Voices is highlighting the research of Plymouth State faculty doing White Mountains and Lakes regions research. If you're interested in seeing some of the amazing past presentations we've had, you can visit the link that Rebecca's about to put in our chat. I also want to thank you if you're a member of the Museum of the White Mountains. Your contributions make exhibits and presentations like this possible. If you'd like to become a member, renew your membership, or simply donate to the museum, you can also go to the link that Rebecca's about to put into the chat. So you now have two links in the chat. One will take you to former presentations, and one will allow you to become a member and support the museum. During this presentation, if you have any questions or comments you'd like to make, you're going to be on mute. So please use the chat function at the bottom. And at the end of the talk, we'll have time for people to ask questions or for us to read the questions to Dr. Yurewicz. So let me introduce Dr. Yurewicz. Dr. Kerry Yurewicz is currently the Helen Abbott Professor of Environmental Studies. Congratulations, Kerry. Kerry was awarded this endowed professorship because of her work as an ecologist whose research interests include animal behavior, species interactions, and patterns of biodiversity across environmental gradients. Most of her work is focused on amphibians and invertebrates, often in freshwater systems. She teaches in the Biological Sciences program. I'd like to add that Kerry uses her connections in social media to share her knowledge and her enthusiasm for the natural world, creating a lovely community of people who are also interested in this. I'll turn it over to you now, Kerry. Thank you.
Wow, thank you so much, Marcia, for that nice introduction. And I will get my screen shared. Just get myself set up here. It's been a little while since I've given a talk on Zoom, thankfully in some regards, but nevertheless takes a little bit of practice to get back used to it. So thanks for that introduction. Thank you for the opportunity to participate in this wonderful seminar series, and thanks to everyone for joining me this evening. So here at Plymouth State, we're currently in the very busy but exciting throes of the end of the semester. But even now, when my schedule is most hectic, there's something about knowing that the salamanders are emerging from their winter retreats and embarking on their ancient spring rituals that is somehow at least a little bit comforting. But I do think that salamanders can be somewhat underappreciated. I'm sure not among this audience, but in general. So it's nice to have some time on this April evening to talk with you about these remarkable animals. And I do have just full transparency. I'm really tired this week, so I thought very carefully about what I wanted to say, and I'm going to be looking at my notes so that I don't ramble off and take you on some wild tangents, hopefully, as we go through this presentation. So salamanders, a little bit of salamander stories before we get into the bulk of the talk. Salamanders overall, I think, don't intrude upon our consciousness as readily as other kinds of animals. And as an introvert myself, I feel a kinship with them for that. Unlike their noisy relatives, the frogs, salamanders usually do not vocalize. So while many species are gathering to mate this time of year, they're not going to be telling you about it, like the peepers and the wood frogs are currently doing for us here in our neck of the woods. In addition, many salamanders are nocturnal, and many spend much of their time in refuge or underground. So you might say they live quiet lives, literally, and in some cases or senses figuratively. There are also many fewer species worldwide of salamanders than frogs. The current estimate that I found was about 767 species of salamanders globally, versus about 8,451 species of frogs. And salamanders also have a much more limited global distribution. So these maps here represent global patterns in biodiversity of salamanders on the top map and frogs on the bottom map, with the warmer colors representing higher species diversity. Note that the scale is different for these two maps, reflecting that salamanders have lower overall diversity worldwide than frogs. And frogs occur on all continents except Antarctica. But native species of salamanders are missing entirely from Australia, as well as sub-Saharan Africa, and broader swaths of some of the other land masses compared to frogs. Personally, I've been fascinated by and have studied salamanders for many years. The main focus of my graduate research at the University of Michigan was studying competition and predation among 3 species of ambistomatid or mole salamanders, their larvae in particular, and their distributions across environmental gradients. One of the most familiar mole salamanders, both where I worked and here in New England, is the spotted salamander, Ambystum imaculatum. Isn't it beautiful? And this species provides a nice example of kind of the typical amphibian life cycle. The word amphibian refers, in a sense, to this double life that includes both aquatic and terrestrial stages, as in spotted salamanders, which deposit their eggs, for example, in woodland pools, and those eggs hatch into an aquatic larval stage that's a stage for rapid growth. During metamorphosis, the aquatic larvae will transform into terrestrial juveniles that leave the water for a life on land, eventually maturing into adults and repeating the cycle, but in the case of the spotted salamander, only returning to the aquatic habitat to lay their eggs. But our protagonist this evening, the Eastern Red-backed Salamander, Plethodon cinereus, is a species with a different kind of lifestyle in a number of important ways. And so when I began working on this species again here at Plymouth State a few years ago, it felt like going home to me in a manner of speaking, because this was the species that was the focus of my first truly immersive research experience as an undergraduate student. And this was at the Mountain Lake Biological Station in Virginia. Never mind how many years ago. I noticed when I found this old photograph of me in the lab at Mountain Lake processing salamanders that my hair has changed color from brown to gray in the intervening period. But nevertheless, the fascination that I developed back then with both the organisms and the kinds of scientific approaches that we used to study them were really pivotal in terms of my path in developing as a scientist and where I wound up. So it was really a huge treat to have the chance to think about what stories I can tell you about this species. And if you didn't know that this was going to be a love story, then, surprise. All right. So with that preface, this evening I'd like to share with you an introduction to some red-backed salamander ecology and behavior, some discussion of conservation challenges or threats facing salamanders like redbacks, and finally, some insight into a population that I've been studying in the White Mountain National Forest. So redback salamanders belong to the most successful family of salamanders on Earth if we measure success through biological diversity. Globally, the family Plethodon today contains 493 species, and that's about 64% of global salamander diversity. And most of that diversity is found in the Americas, with 2 hotspots of especially high concentrations of species, one in Central America, here, and the other in the US, within the Appalachian Range in particular, so along the eastern US. This map is showing the distributions of about 440 species of plethodonid salamanders in the Americas. And the darker colors represent areas where there are a greater number of overlapping ranges of different species. Even right here in New Hampshire, about half the species listed by New Hampshire Fish and Game as occurring in the state are plethodontid salamanders. So here's our gallery of, beautiful gallery of salamanders that occur in New Hampshire. And so we have mud puppies and newts. Here are 4 species of ambistomatid or mole salamanders. And the remaining 6 here on the bottom are all members of this successful family Plethodontidae. Spring salamanders, duskies, 2-lined, 4-toed. Here's our Eastern Red-backed Salamander, and then the Slimy Salamander. I put a question mark by the Slimy Salamander because apparently this species is known in New Hampshire from just one historical record from one town in New Hampshire, so it's not really clear whether it warrants inclusion in our species list. Which personally I find rather sad because that is actually my favorite salamander species on Earth. But nevertheless, plethodonids make up about half of our species in the state. So what are some of the characteristics that help define this group, the Plethodonids, this really successful family of salamanders? Well, some of those characteristics include direct development, the presence of structures called nasolabial grooves, and the absence of lungs. So direct development is a deviation from the typical amphibian life cycle that I mentioned earlier. Rather than eggs hatching into a larval stage, eggs hatch directly into terrestrial juveniles. This life cycle change is thought to be important in the success of this lineage. This is the only family that this developmental mode is found in among salamanders. And while not all Plethodonid salamanders have direct development, the majority of species do. And so you can see in this photo, the adult salamander underneath, there's its eye and its front leg, and it's got these relatively large eggs compared to its body size, where you can see the eye spot of the developing juvenile in that egg and the yolk that it's gaining nourishment from. And when it hatches from that egg, it won't be a larva with gills, it will be a terrestrial juvenile. Nasolabial grooves, another defining characteristic, are furrows that run from the nostril to the upper lip. You can see it here in this picture of a spring salamander. Here's the nostril, and here's this little groove that's running to the top of the upper lip. This is in chemical sensing or olfaction. When a salamander touches its snout to a surface, capillary action moves the odor particles up the groove and to the sensory organs of the nose. And chemical sensing or olfaction are really important to social interactions and locating food, for example. And then finally, and perhaps surprisingly, Plethodonids, this really successful family, are also lungless. This means that all of their respiration takes place across the surface of their skin. Their skin is their respiratory organ, and it has to be kept moist in order to function. Just as we would not thrive or appreciate it if our lungs dried out, a lungless salamander cannot function if its skin dries out. And you can see the sparkle of this really moist skin on this photo of the spring salamander. So they really are constrained by that need to keep their skin surface moist. Even within this very successful family, the Eastern Red-backed Plethodon cinereus is a success story as a species. It's a very common species. It's abundant throughout its range. More on that later. And it has a really wide geographic distribution. So this map shows the range of red-backed salamanders extending from Quebec down to North Carolina and over across the Midwest and even into parts of Minnesota. Within this range, redbacks occupy deciduous forest, mixed coniferous deciduous forests, and sometimes northern conifer forest. By most accounts of this species, their highest abundances in their range occur in mature hardwood forests that have deep soils and abundant downed woody debris, so fallen logs and branches that are in various stages of decay. That is because within these habitats, red-backed salamanders require and spend most of their time in underground retreats, burrows or natural cavities, under cover objects such as rocks or logs on the forest floor, or in deep pockets of the leaf litter. Redbacks are also a completely terrestrial species. They do not occupy streams or ponds or wetlands in any part of their life cycle. And remember again that they're lungless, remarkably, and breathe through their skin. So moisture is really essential to them to obtaining oxygen and surviving in their terrestrial habitat. A little bit about the physical description of red-backed salamanders. So they are small salamanders. Some pictures here of salamanders in the hand for scale. So this picture on the left is a picture of a fairly recent hatchling. So this is a very young juvenile that would have hatched from the egg in that season. And this is by no means a lunker red-backed salamander, but I didn't have a good picture of a lunker, like a really large one, but gives you a little bit of the sense of scale. So the maximum size of a redback is probably about 4 inches long from the tip of the snout to the end of the tail. They can reach maturity in about 2 years after hatching, although it may take longer than that in more northern populations. And they're probably fairly long-lived for what we might think of for such a small creature. In captivity, they are capable of living on the order of 25 years. And in the wild, it's not unreasonable to think that individuals could live as long as, somewhere in the ballpark of 8 to 10 years. Interestingly, and something that may not be evident to us around here, is that red-backed salamanders are not all red-backed. This is a species that is polymorphic or has multiple color forms and patterns. There are several different color forms or morphs, and one of the common ones is called a red-backed salamander. So it's the same species as the classic redback with this beautiful stripe of kind of orangish to red to brown. But this one is a leadback, so it's missing that distinctive coloration. The red striped morph, or the redback morph, is the most common one here in our area. And in fact, in the population that I'm studying, it is the only morph that we've yet to encounter here. But in other parts of the range of this species, the leadback morph may actually comprise the majority of the population. A little bit about redback behavior. There has been an astonishing wealth of research on redback behavior over many decades of research. They do engage in nocturnal activities. So for example, on rainy nights during their most active seasons, when conditions are good, they will emerge from under cover at night and forage on top of the leaf litter, and even climb up on plants and vegetation or the trunks of trees to hunt for their invertebrate prey. They're also a very territorial species, with both males and females defending cover objects from other individuals, sometimes violently. I couldn't resist including this figure from Robert Yeager's foundational paper in 1981, in which he described some of the patterns of aggression within the redback species between individuals. And so this is a figure from his paper, illustrating some of the ways that these tiny yet ferocious creatures will attack each other. So in the top panel, we're going to call this salamander the aggressor, the one that's shaded in. Perhaps it's defending its territory. We don't know its motivations, but that is a good guess. And here's the other salamander. We'll call that one the intruder. Perhaps this is intruding and trying to take over the territory of the aggressor. In the top panel, we see this one taking a nice bite, a chomp on the trunk of this salamander and leaving a little battle scar behind. In the middle panel, we see another aspect of redback behavior. So here our aggressor is taking a bite on the tail of this intruder, and they have the ability to break off or shed their tail without dying, and they can regrow that tail. So that's another possibility with aggressive encounters. And then the final and perhaps most disturbing image and encounter in terms of its sheer ferocity is this last panel, where our aggressor is biting the snout of the intruder. And so they do find, and he did report, injuries where they find bite marks on the faces of other salamanders that can damage the nasolabial grooves, compromising the olfactory ability of an animal that's injured, and that can be devastating to their behavior. So they are fierce and they are very territorial in their season with their cover objects. Chemical sensing is really important to these animals. There's evidence, for example, that individuals can recognize their neighbors around their territories by the chemical cues that they leave in those areas, and that they can even assess the quality of potential mates by the scent of their fecal pellets. Imagine that, by assessing the diet of an individual based on the scent of its fecal pellets and rendering some judgment about their quality as a potential mate. As I just mentioned a moment ago, redbacks have this interesting ability to autotomize or break off and then regrow their tail. Many individuals have blunt or regrown tail tips, as you can see in this picture. And that's either from competitive, aggressive, territorial interactions or failed predator attacks. It's very easy to see. So in this picture, we see the clear distinction between the original portion of the tail and then the regrown portion here. And part of why that's so visible is that they regrow the length of their tail prior to regrowing the width of the tail. So it makes it very distinctive to look at. And then finally, in terms of behavior, another really interesting aspect of their behavior is parental care. Female redbacks will brood their eggs for an incubation period lasting 6 to 8 weeks, roughly. And during this time, the female, the mother, will not leave her eggs to actively forage. So this is a really energetically costly investment in her young. She is coiled around those eggs and will protect them from predators as best she can and keep them moist during their incubation period. During that period, as I said, they're not out actively foraging. She's not leaving the eggs to go hunt for prey. She will opportunistically eat what may come by the area where she's guarding those eggs. But other than that, she's really reliant on fat reserves in the body and fat reserves that they store in their tails to basically help them withstand this really stressful breeding period. So what does a year in the life cycle of a redback population look like? If we think about that through the seasons, now we're in springtime, the weather's warming up, we're getting some nice spring rains even this evening, and animals, including redbacks, and even some other animals are emerging from their winter retreats. Red-backed salamanders would be establishing their territories, coming up from underground and looking for cover objects to occupy. They'd be certainly foraging for invertebrates that are starting to become active on the forest surface. And so we expect to see in areas like New Hampshire, northern areas, this kind of surface activity peak. Conditions are good for them in the spring, and they tend to be a lot of individuals in the population up on the surface. As spring transitions into summer, females that have mated will be laying their eggs and brooding their eggs throughout the summer season. It may get too hot and too dry, so surface activity tends to be lower in the summer overall, and many animals will be spending more of their time underground where conditions are cooler and moister. And females will guard their eggs until they hatch in late summer, maybe August to September in our area. As we continue on to fall, then temperatures get cooler again and a bit more favorable for surface activity. And so we tend to see another surface activity peak where we have more individuals in the population active and visible, undercover objects, but up on the surface or in the leaf litter. This is probably when most mating in red-backed salamanders occurs, although it can also occur in the springtime. And then in the winter, unlike some amphibians like wood frogs that can tolerate periods of completely freezing, red-backed salamanders cannot. So they need to go underground, and in fact, they need to be below the freezing line. So they have to get down to a level of the soil where they're not going to be exposed to those freezing conditions. So we talked a little bit about their behavior. Now in terms of their ecology and where redbacks fit into their food chain, redbacks have been reported to eat a wide variety of different kinds of invertebrate prey, including things like ants, termites, flies, earthworms, snails, mites, caterpillars, slugs, basically anything that they can successfully capture and ingest. They in turn have a variety of predators that will feed upon them if they can. That list includes species like garter snakes, shrews, raccoons, centipedes, some large ground beetles, and even some birds that forage in the leaf litter, for example, are some of their potential predators. So based on these observations, they can influence the energy flow in their ecosystems. By preying on small invertebrates, they have the potential to influence the dynamics of soil and leaf litter communities and the decomposition or decay rate of that leaf litter on the forest floor. They're also an abundant and high protein source of energy for predators higher on the food chain. And so the impact of red-backed salamanders in their ecosystems and communities may in part be substantial because they reach such high abundances. And estimates of the surface density of red-backed salamanders across their geographic range in the United States have ranged from about 0.05 to more than 3 per square meter. And there's recent evidence that in some places may be even higher than that as we obtain more data. I want to tell you a little bit about a famous foundational study about red-backed salamander abundance in New Hampshire that was done by Thomas Burton and Jean Likens, published in 1975 from work done at the Hubbard Experimental Forest just north of us in West Thornton and Woodstock. So this map, here's I-93 heading north, and here's the boundary of the Hubbard Experimental Forest within the White Mountain National Forest. And Burton and Likens were working to estimate the abundance of many species of salamanders that occur in the Hubbard Brook experimental forest. And they did this work in the summers of 1970 through 1972, including estimates of red-backed salamander abundance in watershed 6 and 4. And so those are labeled here on this map. Here's the area of watershed 4, and here's the area of watershed 6. And they used two different methods to try to get a handle on how abundant redbacks were in these areas. So they did surface census plots, and these were all done during the summer. And so on summer days, on dry days, they would have these plots, square plots, 25 by 25 meters on the sides of known areas that they would have distributed in their sample areas. And in these known plots, they would go through and look under all the rocks, under all the logs, and in deep pockets of leaf litter and count up how many salamanders they would find within those defined areas and take multiple plots of that type. They also combine this with transects on rainy summer nights. So to capture that, estimates of their activity, their nocturnal activity on rainy nights. These transects were each 240 meters squared, and so that would involve setting up these transects and walking along them and looking for salamanders that were active at night in these rainy conditions. From these kinds of methods to estimate the abundance of salamanders, they then took into account many factors to try to extrapolate from their subsamples to estimate, for example, across the watershed, how many total salamanders they were and how significant they were in terms of the biomass and abundance. So for example, they factored in that there were elevation differences across the area that they were sampling and considering. They factored in and considered published estimates that only 2 to 32% of redbacks in a population may be visible on the surface at any given time. They considered that during the summer, when they were sampling, females with eggs certainly would be underground and not counted because they wouldn't be visible. So taking into account many of these factors and others, they arrived at their estimate as best they could for watershed 4, factoring in lots of these things. So their estimate for watershed 4 was a total of 2,583 redbacks per hectare, summing up to about 1,650 grams of redback salamander tissue per hectare, so biomass, or just amount of wet mass. And they noted that because they were surveying for other salamanders as well, the redbacks accounted for 93.5% of the total salamander biomass in their samples. Now, I've always had this weird mental block with the hectare. It's something I just can't, for whatever reason, wrap my head around. So I thought, what would that translate into something that I could relate to a little bit? So I decided just to do some arithmetic and relate it to the standard NFL football field. And so if we do that, think about that area, taking their estimates per hectare, what would that translate to on a standard football field? We would get, if I did my arithmetic correctly, always a question this time of year, 1,382 redback salamanders on the football field, and that would sum up to about 2 pounds of biomass, which doesn't sound super impressive, I guess, but remember that these are really tiny organisms, so that's impressive in its own right. But what was more and truly impressive was that Burton and lichens could compare their estimates of redback salamander abundance and biomass to other well-known vertebrates of the Hubbard Brook ecosystem. And this is what made this paper make such a splash, one of the reasons it made such a splash. So by their calculations, their estimates of abundance and biomass, there were more salamanders at Hubbard Brook than either birds or small mammals in terms of numbers. And the biomass of salamanders at Hubbard Brook was higher than the biomass of all birds during the peak of the breeding season by 2.6 times. The biomass of salamanders was about equal to the biomass of all mice and shrews combined. Again, so clearly, and just to remember, redback salamanders, this one species, made up 93.5% of that biomass. So this was the species that was really contributing to that huge abundance in biomass. So it's clearly one of the most common vertebrates in northeastern forests here in North America and elsewhere across its range. However, more recent estimates and observations challenge this picture of redback abundance at Hubbard Brook and elsewhere. So Richard Holmes and Jean Likens in 2016 published a book about the Hubbard Brook ecosystem. And in that book, they mentioned that they had done that surveys that had been done in the same areas that they surveyed back in the 1970s, but more recently, from 1995 to 2006, using the same methods in both periods, yielded many fewer red-backed salamanders. So their estimates of density, for example, in watershed 6, went from 0.26 redbacks per meter squared in the early 70s to 0.075 redbacks per meter squared in 1995 to 2006, a decline in abundance of 75% during this 25 to 30-year time span. That would be like in the 70s, if you sampled 4 square meters, then 1 out of 4 of those square meters you would find a salamander. If you went back in 1995 to 2006, you'd have to sample 13 square meters to find that one red-backed salamander based on their lower abundance. Elsewhere in the range of redbacks, others have also noted declines. So for example, Heighton published in 2003 a summary reporting on his efforts to revisit a bunch of sites, 24, in a variety of different states that he had surveyed in the pre-1990s and then revisited again in the 1990s. And he reported that by his estimates, the average catches of redbacks had dropped by about 50%. Now, it is important to note that others have published studies where they have reported no indication of declines in other areas. So, for example, there was a paper that was published in 2017 where they were assessing redback abundance in Ontario, and they saw no indications of decline. So that population abundance seemed to be stable. So there's lots of open questions here. What do these observations of declines mean in places where that has been observed? What could be driving these patterns? Is this a widespread pattern? And what might it indicate about the environments that these salamanders live in? So with that, I'd like to talk briefly about some of the challenges facing salamanders in terms of their conservation. Now, we know that salamanders are an imperiled fauna worldwide. The IUCN, the International Union for Conservation of Nature, estimates that 40% of salamander species globally are endangered or critically endangered, and another about 17% rated as vulnerable. The threats to salamander biodiversity are the same kind of usual suspects in many cases as threats to other forms of biodiversity, so including habitat loss and modification, pollution, invasive species, emerging diseases, climate change, and more. But how do these threats relate to red-backed salamanders in particular, or how might they, and what are we learning about that? So just a few comments about that. In terms of habitat loss, red-backed salamanders thrive especially in mature forests with an abundance of moisture and woody debris on the forest floor. So certainly development and conversion of forest into other landscapes can threaten red-backed salamander populations, as well as fragmentation of forest into smaller patches that can change the condition in those patches. Forest harvest and management of varying types, different areas can create local conditions that are less suitable for red-backed salamanders, at least for a while. There is evidence that populations can recover and rebound after even clear cuts if those areas are left to regrow and ecological succession takes place. The red-backed salamanders can come back, but that's certainly a change. There are also forest pests. This is another, also tying into introduced the non-native species, but forest pests that may alter the properties of our forest ecosystems. So for example, the hemlock woolly adelgid is an invasive insect seen in this picture here. It's the white blobs on this hemlock that is causing declines of hemlock in hemlock forests in the northeastern US. So there have been some interesting studies that have come out of Harvard Forest in Massachusetts, where they had plots where they girdled hemlocks to simulate the impacts of invasion. And these experimental manipulations resulted in changes in soil qualities, such as temperature and pH, in those plots that were associated or correlated with decreased red-backed salamander captures. And that's just one example. So ongoing research to try to understand what some of these changes might mean for redbacks. In terms of pollution, the impact of contaminants like pesticides, herbicides, or salt runoff, and others have been studied much more extensively for aquatic amphibians and their life stages than terrestrial amphibians in many cases. There was one interesting study from Ohio that demonstrated that red-backed salamanders showed behavioral avoidance of 3 different herbicides when used at their full recommended application dosage. Other researchers have concluded that redbacks are good bioindicators of mercury bioaccumulation by looking at levels of mercury deposition and mercury accumulation in the tissues of salamanders and other organisms in their ecosystems. And for one last example, for acidification or acid rain legacy, early research on redbacks suggested that very acidic soils with a pH of 3 or 4 negatively impacted the growth and health of their populations. Although a more recent study in New England found that they may be more tolerant of high soil acidity, low pH, than originally believed. So there may be some adaptation or greater resilience or tolerance than some of those early studies were observing. In terms of invasive species, I can give you an example of one active area of research with red-backed salamanders has been investigating the connection of invasive earthworms to redbacks and their decline. Through their impacts on the leaf litter and soil communities, invasive worms can impact the invertebrate prey that red-backed salamanders rely on. So you may hear news stories about crazy worms and jumping worms and all manner of different kinds of non-native worm species that have been introduced to different places. And there's been a lot of research going on to try to understand how this may impact those leaf litter communities, including red-backed salamanders. Emerging diseases, new diseases are not just an issue for us, but also a major threat to other species. Many species of amphibians were devastated by exposure to chytridiomycosis, which was a novel fungal disease here and in other places around the globe, so not a non-native species that was introduced. As it turned out, red-backed salamanders were not particularly susceptible to chytridiomycosis, so that was in their favor. But there is a new fungal pathogen, also a chytrid, that is causing great concern because it reached Europe and caused significant declines in fire salamander populations. That's that gorgeous animal picture at the top. The fungal pathogen has been abbreviated as B-sal, and that's coming from the scientific name of that fungus. And agencies like the USGS are actively monitoring amphibians here to test for the presence of this new disease and to try to understand what the impacts may be if or when it reaches North America. And they think that it could be severe based on what they observed in Europe and what they saw with chytridiomycosis earlier. And then last but not least, climate change may have many impacts on the distribution and success of red-backed salamanders. So there's a data set of more than 60 years of data from the Hubbard Brook Experimental Forest that indicate how our climate is changing right here in the region, in the Northeast, and that includes warmer average annual air temperatures, increased precipitation, and decreased snowpack. All of these changes can affect red-backed salamanders. Temperature through their metabolic rate, precipitation via their critical need for moisture to keep their skin moist, snowpack through their need to take refuge below the soil freezing line in the winter. There's just a wonderful kind of one-page PDF, or two-page PDF version, Hubbard Brook publication about facts about climate change that summarizes lots of these trends. So I encourage anyone, if you haven't seen that, to check that out. Really nice summary of some of the changes that are coming from what they've observed in these incredibly long and detailed data sets. And so as one approach to investigate how climate change may impact red-backed salamanders, I've been involved in a regional research network studying this species across its distribution. Our research network that's shown in this map shows the geographic range of the red-backed salamander in the blue shading. and then research and observation sites of different types with these little red and yellow markers. The red markers indicate sites like the one I'm going to tell you about at Hubbard Brook, where we're doing a capture, mark, recapture method to estimate abundance and other aspects of red-backed salamanders. So here's our Hubbard Brook site right here. So since we don't have a time machine to go forward another 60 years or 100 years and see how things worked out for red-backed salamanders with climate change, we can use a space for time approach, meaning we can look at different locations now that have different climate conditions and try to build an understanding from that about how populations will change as climate conditions change. And that's what we're attempting to do with this broad collaborative research network. As part of this network, we're using a standardized method across the range of redbacks to collect data, hopefully long-term, on abundance, aspects of population structure, growth rates, and behavior in different parts of their range. So let me share with you a glimpse into what we do and what we're starting to learn here at our study site in the White Mountain National Forest at Hubbard Brook. And I have to admit, it says every scientist ever probably, or at least me, I am not up to date with getting all of the data entered. So goals for the future, but I'll share with you some glimpses of some of the observations that we've made so far. So it may look odd to see a grid of small square-foot wooden boards out in the forest floor, but this is actually a common method for studying redback and other small terrestrial salamanders. So this is one of our arrays of what are called artificial cover objects. So it's a 5 by 10 meter grid with 50 cover objects in it, arranged in a rectangle. We set up at each site arrays of these boards, and by clearing out the natural cover objects from these sites when we first establish them, so clearing out any down logs or rocks that are movable, and arranging this grid of boards, we create an array of artificial objects that salamanders will readily colonize. This standardization of the array size, the number and spacing of the boards, allows for a fair comparison of the populations that we're looking at across different locations than if we were looking at areas that had very different numbers of cover objects or type of cover objects that were natural. Each board in an array is individually numbered so that we can identify the exact location where each salamander was observed. So we use what's called a capture, mark, recapture method to estimate abundance of our population. And when we survey a plot, we systematically visit each board in the plot in the array, gently lifting it up and looking for salamanders. Doesn't look like there was anything under that one. But if we do find a salamander, any salamander under a board will be gently collected and placed individually in a Ziploc bag that is labeled with the board ID number as well as the plot where it came from. And then we just replace the board to its position. That's the capture part of the capture, mark, recapture survey. Once all the salamanders from a plot have been collected, we then take them to a laboratory space for processing. And there we measure each individual. We measure their total length from the tip of the snout to the end of the tail. We do have to get them to line up as straight as possible for that measurement to be accurate. Hopefully we do a little bit better than in this drawing. And we also measure a standard herpetological measure called snout vent length, which is from the tip of the snout to the anterior end of their vent, which is their reproductive and excretory opening. And the reason why we measure snout vent length is because since these animals can break off or shed their tails, if we just measured total length, that could give us some really wildly ranging estimates based on an animal with a very short tail that's broken or an animal with a fully grown tail. Whereas snout vent length estimates of length should not be affected, are not affected by that. So we measure them for their size. We then use a flashlight to do what's called candling the salamanders. And all that means is just because their bodies are so small, when we have a light shining through underneath them, I can tell if a salamander has testes, which would indicate that it's a mature male, or I can tell and even count if a female has eggs in her body, which would be definitive evidence that it's a mature female. The first time an individual is captured from the plot, we mark it with a unique code of tiny spots of what's called visible elastomer, a fluorescent silicone-based substance, just under the skin at the base of the legs. There are 4 different colors of elastomer that we use, and up to 4 different spots under the base of each leg that we can mark an individual salamander. And this yields a huge array, a huge combination of different unique ID codes that we can assign to different individuals. So this too is a standard method that has been used to uniquely identify individual amphibians in many research studies. So for example, if this salamander were marked with the colors and positions shown, its unique identification based on blue, orange in those positions would be Bobo. And we can hopefully identify Bobo if we capture Bobo again on a later date. This is a photograph of one of the salamanders. So these visible elastomer marks are most visible under a black light. And so this one has no mark under this arm. It has a red mark here. Hopefully you can see that. It's not the greatest picture. There's a blue mark here and a yellow mark there. So this individual's code would be no mark, X, and then red, yellow, blue. So XRYB for that individual. Following their alien abduction experience, all salamanders are returned to the exact board that we found them under. So that's the capture and mark portion of capture, mark, recapture. The recapture part means that after the very first visit to an array, when every individual is new to the study and is marked, we will visit a site again, and then we'll expect to see a mix of new individuals that we didn't see before, as well as recaptures, salamanders that were marked previously. And so if you have a very simple case where you have 2 visits to a site, just an initial visit and a return visit, we can use this basic equation to calculate or estimate the abundance of animals in the plot, where N is the abundance of animals in the plot, and we need to know the number that we marked the first time we visited the plot, the total number that we caught the second time we visited, and we divide that product by the number of animals that were marked in that second visit. So let's say we went out to our plots on date 1 and we captured 10 salamanders and we marked them all. And then we return later, a couple weeks later, and maybe that's a really busy day, and we caught 30 salamanders, but only 2 of them had marks from that previous visit. So those would be our 2 recaptures. And so if we plug those numbers of 10 marked the first time times 30 the second time divided by 2 recaptures, we would get an estimated number of 150 animals for this plot. Notice how much bigger that is using this mark recapture method on this low number of recaptures in this example, compared to what our estimate would be if we just looked at surface activity. So if we just looked at surface activity, we'd say there were 10 salamanders based on this visit, we'd say 30 on this visit, but our mark recapture method gives us an estimate that is based on the knowledge that not all animals are visible on the surface at any given time, and so we're going to have this mix and movement of animals within our plot. This basic equation can then be modified for cases where we have multiple visits to a site, not just 2, which is what we're doing, as well as where we have spatially explicit capture data. So we know not just how many salamanders we captured, but exactly where we found them under individual boards. So just a few glimpses of data as I try to wrap things up pretty quickly here with the time that we've got. So how abundant are red-backed salamanders at this site in the Hubbard Brook Forest that we've been working at? So if we just look at surface activity, so not the capture, mark, recapture, but just the animals that are on the surface when we visit in the spring and the fall, we see on average about just under 10 per plot on average, which works out to an estimated density of about 0.2 per meter squared. And that's not terribly different between the spring and the fall, and we do sample during these peak activity times. If we compare that to our estimates from our mark recapture process, now we have our surface activity estimate of around 0.2, much lower than our estimate of the actual density of the population, given this knowledge that animals are not all visible at any given time. It's not quite 10 times as high, but it's closer to 2 per square meter based on that method. And I always think of it as like sort of this molten layer of lava underneath of salamanders that are under the surface. And so as salamanders move and change their behavior and go about their lives, there's always like new salamanders or different salamanders burbling up from below. And even on really good days when we take salamanders back to the plots to release them under their boards, Sometimes we'll find new salamanders that have come up from underground during the time that we've been measuring others to occupy those artificial cover objects. It's really remarkable how abundant they are. A few quick observations about some other population attributes. We've been keeping track of the number of eggs or the clutch size that pregnant or gravid females have. And the mean and median are around 6 for our observations so far. And that's well within kind of the published range of clutch sizes that are out there in the literature. We do see about 20 to 30% of our observations of animals have signs of tail damage. So this individual has a shorter than expected tail. And you can also see that tip that is actively been regrown here. So it's still working on regaining its full tail length. So we see a lot of evidence of competitive interactions or predatory interactions that result in tail damage. And we're also looking at the distribution and movement of juveniles versus females versus males. So in this case, I've just summarized some simple data for juveniles versus adults to show you that during our observations in the springtime tend to be more highly weighted towards juveniles than adults, where it's about more evenly distributed in the fall. So we're starting to look at some interesting patterns of what different life stages of salamanders are doing. And so the last little bit of data that I'll share with you is one of my favorite things about red-backed salamanders is their remarkable site fidelity as highly territorial animals. They are highly likely to remain in their core location even after a disturbance like their experience being captured and measured and candled and all of this kind of thing. So we looked at data for this graph for just the first time, a bunch of observations of just the first time we recaptured a bunch of salamanders. And so if we have their location where they were first captured, let's say under board A1, and we have their location the first time they were recaptured, let's say it was board B1, then we don't know everything that they did or their movement in between those two times, but we can at least say a minimum distance that they would have had to move to occupy this new location the second time we captured them. And so we can calculate that, or just this is about 1 meter, this is about 1.4 meters. So anywhere in that plot, we could calculate at least the minimum distance they would have to go. And PSU student Ellie Wilson has been compiling some of this data by crunching these numbers. And so this graph shows the percentage of individual salamanders moving different distances from the time of their first capture to their first recapture. And you can see that the majority of animals do not go anywhere. If you find them under board A1, then the next time you go back to recapture them, they are in the same place because that is their territory. And even having been disturbed, they are going to reclaim that territory and maintain it. About 30% may move one board over, so not very far, and probably within the home range area, kind of core area of the red-backed salamander in any case. And it's very rare for them to be recaptured at sites that are further distant from those areas. They really are very faithful to their site. If we look at kind of how long was it between their capture and first recapture, we see that that can affect the distance we see between their capture and recapture sites. So this is minimum distance moved, like for animals that we recaptured within a season, so say within the same fall season or same spring season. Between seasons, so maybe an animal we captured first in the fall and we didn't see it again until the following spring, or between years, so maybe an animal that we didn't see for 2 years between its first capture and its recapture. And so you can see that the minimum distance estimates go up, but you can also see the scale that even on the high end, it's still a very low distance. They're very faithful to their sites and their territories, even between years. And so just a couple of anecdotes about salamanders. So the salamander XOXB is a female. We have observed her 5 times over the years at board D9. That was her spot in her plot. Over 4 years. Every year we go, she's always back there in the same place. XBXR is another female. She was initially observed in 2017 at board B4 in her plot, and we didn't actually see her again for 4 years until 2021 when she had moved just 1 board over to A4. So it's really quite an interesting, some interesting data that we're going to be able to dig into about their movement. Right, so now that we're accumulating some really solid data sets through this regional network, really excited about and looking forward to using the power of this collaborative research network to look at differences between sites, correlations with environmental variables, to understand patterns in growth, abundance, and density, and relate them to climatic variables. And also other ideas for looking at prey abundance in different locations, looking at their population genetics. There are a lot of moving parts to this research network that will hopefully keep me busy for many years to come. With that, I'll conclude. I'd like to just acknowledge the museum for inviting me to participate. I'd like to acknowledge Plymouth State University for support for this project over the years. I'd like to especially acknowledge my student research assistants, especially Liz Service, Emily Blakey, Ellie Wilson, Dave Wynant, and Adam Bowman. I'd like to thank the Hubbard Brook Research Approval Committee for permission to work at the site. The U.S. Forest Service and the Hubbard Brook Research Foundation have both provided me access to lab space to measure and process salamanders, and also to my research network collaborators, especially Evan Campbell Grant from USGS, who recruited me to this network, and Jill Fleming, as well as a colleague, Caitlin Fisher-Reed, who's down at Bridgewater State in Massachusetts, who's been a very valuable source of advice as we've been working on this project. So with that, I will stop talking and will be happy to answer any questions as best I can about red-backed salamanders. So thank you for coming along for this ride with me.
Yeah, agreed. You see the hands up and the cheers and the clapping. Ditto. And I had never known redbacks were so daggone cute.
That's the easiest thing to learn about them, yeah.
Let's see, let me read some of the questions you have. You have quite a few. Did you use the word mud puppies?
I did use the word mud puppy.
What's
yes.
a mud puppy?
So a mud puppy is a particular species of salamander. That's just their common name, mud puppies. I believe, yeah, the genus name is Necturus, but they're aquatic salamanders. They're pretty big. I've never seen one myself alive. I've seen pickled ones, but yeah, they do occur in the state. I'm thinking that's probably in like the Connecticut River or something like that, but I'm not an expert on mud puppies. But yeah, that's their common name. Pretty cute.
It's a very cute name. I'm apparently on a cute kick tonight. Both of those were my comments. Is it possible, Amy asks, for the soil to be too wet for red-backed salamanders? On the flip side, how do these salamanders respond to periods of drought?
Yeah, so yes, so for, when thinking about precipitation patterns, right, critical that there be enough precipitation because they need moist conditions. But the Hubbard Brook data set, those 60 years of data have also shown an increasing frequency of heavy precipitation events. And so areas that get inundated with water or get flooded can also be a risk to red-backed salamanders as they're spending a lot of time in those underground retreats. So that can be problematic. And there have been times out at our plots at Hubbard Brook where some ends of the plots have been flooded after really heavy rains. So don't have enough data to really know what that's doing at the sites that I've been looking at, but it's something that we're keeping track of to see if we see any kind of signals of does that affect our capture rates around those times. Drought periods are definitely stressful for red-backed salamanders. So they just need to go underground to get to moisture conditions when it's too dry. And that limits their activity, that limits their ability to forage. And if they can't find those conditions that are moist enough, then they can easily dry out. So that is a real risk of desiccation is a real threat to their survival.
Someone asked, you may have answered this, but I don't remember, so I'm going to ask it again. How long is the incubation period?
That females are brooding their eggs? Yeah, about 6 to 8 weeks. Variable across the range, but something like that. So, it could be a good 2 months that they are really coiled around their eggs and protecting them. And that's, in fact, one of the things that I worked on when I was an undergrad back at Mountain Lake was looking at the interaction between tail loss and the energy that they need to brood during those brooding periods. So it's quite amazing that they have that level of parental care And females can really lose a lot of weight during that breeding season, especially if they don't have fully grown tails and fat reserves in their tails to rely upon. So they're good moms. They're really tough moms as well. The males are not helping out. I just have to say that, you know, they're not involved, but the females are taking care of the eggs.
So the fathers just disappear, huh?
Fathers disappear. But just to be fair, right? Fair and balanced. Females will also, if it gets too stressful, they may cannibalize their own eggs and just decide to try again in another year. That is also an option that may happen.
So Becky Moore had asked about the Asian jumping worms, and you touched on that. Is there anything additional you'd like to say about invasive worms that you haven't already?
Yeah, I wish there were, but I wish I knew more about it. But yeah, I don't. So there have been a few studies within a geographic range of redbacks looking at earthworms of different sorts, and those jumping worms. Yeah, I came across a few papers, but I'm not an expert in that. So there are a couple studies that seem to suggest that the invasion of those kinds of species could be problematic for redbacks, but I don't know a lot of the details. I think one of them was a study in New York, so not too far away from here.
For those of you who are on, Rebecca Inman put in the chat the Hubbard book, Climate Change Facts. It's really cool. I looked at it a little bit. Lisa Scott asks, what role do vernal pools play?
For redback salamanders, not much. So they are not reliant, they are not using aquatic habitats at any part of their life cycle. Certainly, you can find them around. There would be moist conditions around a vernal pool that may be favorable, but sometimes those habitats near vernal pools are more likely to be occupied by other salamanders, like spotted salamanders, that may compete with and maybe even exclude redbacks from around those areas. So they're not really, redbacks aren't associated with those habitats. They certainly could occur there if they can compete there, but you're not going to find them in there particularly associated with them. But they are really important habitats for lots of other species, including spotted salamanders and wood frogs as well. So, and they're fascinating, and they're humming this time of year, so they're really cool to check out if you have an opportunity.
Do you? So it looks like you've already had students out looking for redback this spring.
Not this spring, but yeah, not quite. We're hoping to gear up pretty soon, but yeah, it's been tough to squeeze it in yet with the semester still underway. But we'll get started pretty shortly, I think.
Then I'll skip one of the questions here, which was mine, so it's okay. I'll catch you at some other point. Becky Noel asks, how do they have occasion to get aggressive with each other, given that they can barely travel from their sites?
Yeah, so yeah, they barely travel. I mean, they do move around a little bit in those very constrained areas. They are foraging, right? So they're moving around to forage. Juveniles also generally, aren't holding territories necessarily of their own. It takes a full-grown adult to maintain that territory. So juveniles often are more like floaters in a population that will be traveling around and may challenge a territory holder for a territory, or maybe one territory is better, one board is better than another, or one cover object is better, and so they may attempt to move into a territory that has a greater abundance of invertebrates. So yeah, I mean that intense territoriality, that aggression, those terrifying images that I hope will haunt your dreams at night as redback salamanders. Help maintain those boundaries and probably limit some of that movement. But there is still activity, and animals coming up from underground, from underground retreats and moving up and down. And so they may not come to exactly the spot they were before. So they do have these opportunities to interact with each other. But yeah, that's a really good question.
Kerry, you have quite a few thank yous, great information, really informative. A lot of people thanking you for your presentation tonight, and I joined them in doing that as well. Thank you very much. I hadn't given salamanders a lot of thought, and now I will, especially when I see them outside. I want to mention to everyone that right now the museum and also down at the Silver Center, the Bachelor of Fine Arts exhibits are up, and I hope you have a chance to go and see them. And also prepare yourself for a climate change exhibit that's coming this summer called Watching the Seasons Change. It'll be at the Museum of the White Mountains between June 4th and September 17th, and we hope to see you there. This is the final Mountain Voices for this academic year. Look forward to seeing you around campus, or in the museum. Thanks, everyone.